This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, a special episode recorded on August 20th, 2019. I'm Vincent and you are listening to the podcast all about viruses. This is TWiV episode number 2 from Stockholm in Sweden. We are back at Karolinska Institute. I can't even say that one right. Karolinska, except we're on a different campus. We're on the Solna campus, and there are many buildings here, hospitals, laboratories. I saw the building where the medicine Nobel laureates give their addresses, and I saw the little building where they announce the prizes. And that's the closest I will get to any of those. And today, I have 3 more guests gathered here by Kim Vesto, who arranged this TWiV for me, and they're all at the Karolinska here. Let me introduce them. To my left, they're all professors here, of course. Jan Albert, welcome to TWiV.
Thank you.
Thanks for doing this. Next to him is Peter Brodin. Welcome.
Thank you.
I prefer to say the names the right way.
Very good.
But I understand why you wouldn't, because everyone messes it up, right? Everyone calls you Peter in the U.S.
But you did well. You did well.
Peter Brodin. And next to him, Anna Smed-Sörensen.
Yes.
Welcome.
Thank you.
It's a good way to say it,
Yeah.
Sörensen.
Yeah, perfect.
At this morning's TWiV, I was corrected several times.
No
By
need.
various people, right? And we have an audience of two people here. We have a long-term TWiV listener, 11 years, right? Amazing. She's a student in Anna's lab, is that correct? PhD student, welcome. And Agnes is here, who was my host for this morning's TWiV. So what I want to talk about your science, of course, but first I want to find out a little bit about your history, where you're from and educated and so forth. So Jan, let's start with you. I assume you're from Sweden, right?
Yeah, I'm from Sweden. I grew up in Stockholm and I did my studies here and my research. And this goes many years back now. So I'm a medical doctor. So I did my training in medicine here. And actually this morning I was at a meeting, or until now, a meeting on how to reorganize the medical education here at the Karolinska. We're going to have a big change actually. So I did my education in medicine here, and I thought that I would be a clinician. So I started out working a little surgery, and then infectious diseases was my real interest. So I started doing some sort of beginning specialty training in infectious diseases. And then I did my the compulsory internship that we have to do here in Sweden to become licensed doctors. And I came back to infectious diseases, and then I had to do some lab training to get my specialty. And it just by chance, really, I happened to end up at the start of HIV research in Sweden. And I was fascinated, and it was a really good chance to start research, which I hadn't really planned earlier. So I was relatively sort of far into my career before I started doing research. And before then, I had also seen the first HIV patients die here in Stockholm, which influenced me quite a lot in my career decisions. And then it's been research and also some teaching and translational research.
So that would have been the 1980s, roughly, the age?
Yeah, I started in the infectious disease department 82, and then was back in 85, and then I started at the public, the equivalent of the CDC in Sweden in 85, late 85.
It's interesting. I've spoken to a number of MDs who have a similar story. They were taking care of patients. All of a sudden, AIDS came along, and then they decided to do some research. So do you still see patients?
No, I don't see them, but I do a lot of translational research, and I'm involved in treatment guidelines and stuff like that. So,
Right.
so I do. I'm still very much connected in patient work, I would say.
Can you remember how far back in your life you wanted to be a doctor?
It was sort of on my list from very early. I knew that I wanted to do some natural sciences.
Right.
But I was also thinking about engineering and also some other things. And then partly, again, by chance, but maybe not in reality, I ended up starting doing medicine.
Okay. Petter, where are you from?
I'm from here as well, Stockholm.
Yeah.
Yep. And I studied medicine also here at the Karolinska Institute. I did an MD PhD program, although at that time there was a program called MD with research alignment, which was basically a way to try to lure MDs into research. It's not, I don't think it doesn't exist anymore here. But anyway, so I did that because I was curious about it. And then sort of accidentally ended up in this field of immunology where I am right now. By virtue of a brilliant lecture by Claes Scherre, who is a sort of pretty famous immunologist and professor here, who teaches immunology, actually still, to the medical students. And he gave a phenomenal lecture about this really intricate system that got me hooked. So I started doing research in parallel with my medical studies. This was in the second semester of medical school. And then I sort of went between the lab and the classes for the rest of my medical school. I actually finished both of them at the same time with a PhD and an MD in 2011. And then I knew I wanted to see patients. I absolutely loved it, especially children. Pediatrics was my favorite course. And now I am a pediatrician here at the hospital, dividing my time between clinical work and then research in immunology, specifically, at least more recently, in children, pediatric immunology.
It's very common that a good teacher
I think so.
inspires us,
Yeah,
right?
right.
That's what I hope to do with these podcasts, is inspire people.
No, for me, it was transformational, completely.
Now,
I did a postdoc also, I should mention that, because that's also been very important to me, shaped my sort of view of things, together with Mark Davis at Stanford University. And that was in 2012 and 13. And that also was a phenomenal experience that really sort of set my trajectory for where I am right now.
he's a good immunologist to talk to, right?
I would say so, yeah. He's a pretty phenomenal mentor as well in science in general.
So when I was looking for a faculty position, early 1980s.
Oh, yeah.
So I'm at Columbia, but I had an offer from Stanford from his department.
Great.
I decided not to go. I decided to stay in New York.
Yeah.
But I might have seen you there. Who knows if I had gone there?
Could have. It's a pretty great place to be.
Yeah. How far back in your life do you remember wanting to be a doctor?
I mean, not far at all, actually. I had completely different dreams when I was a kid. I was playing hockey, and I was going to be a professional ice hockey player, and I loved music. I was into that. And then, again, one of these accidental meetings when I was doing my military service here in Sweden, I knew I liked this idea of being challenged and trying to strive to sort of, push myself all the time. That's what I was doing in sports and so on. And then there was one of these officers I did a pretty demanding military service. And one of these officers there said, well, I've done this now for a few years, but now I'm going to take on a real challenge and I'm going to be a doctor. And so that sort of triggered something in me. I said, wow, that's a real challenge. Maybe I should try that myself. So, that's what it is.
It's interesting that you listened to him, right? That's what the military does, right? You listen to your superiors.
I guess so, yeah.
I mean, that's the whole point. You have to listen if you're given an order or something. If you don't, there's no point in being in the military. They say, go out there and shoot, you have to go. So he said, go to medical school.
He said, basically, you want a challenge?
Yeah,
go to medical
great.
school.
I like that.
And I
really
thought
good.
that was pretty...
Was he right?
Yeah, I think so. I'm still challenged every day, Patients that are very complicated or research questions that are really difficult to answer. So, yeah, I think so.
I should say that My interest in infectious diseases came from a really good sort of course with terrific teachers when I was in med school.
Well, my interest in virology came from a book, and one of our audience members was also inspired by that book, Fever, by John Fuller, right? It's a cool book. I also had good teachers. Anna, where are you from?
So I'm from Örebro, which is another Swedish city, 2 hours from Stockholm.
Is it north or south?
It's towards Norway, so it's west of here. But it's quite a while now since I lived there. I moved to Uppsala when I was 18 to go to university, so that's where I did my undergrad. And then I came to Stockholm for my graduate studies as a PhD student that I did here at the Karolinska, but I also did half of my PhD at the NIH in Bethesda with Rick Kaup at the Vaccine Research Center.
Right. That was a pretty new place.
It was pretty new then. But
I remember when it was started, yeah. And did you know the HIV structure guy? What was his name?
Peter Kwong?
Peter Kwong,
yeah.
Yes.
No, they were a pretty... I mean, I was, of course, very peripheral in this... in the building, but they were a tight group of scientists that were clearly recruited for a common mission. So my thesis was on HIV and on the role of dendritic cells in HIV. So I went there to learn how to isolate primary dendritic cells and then infect them with HIV and study them.
And then, so you did half of your thesis there and half here.
Right.
And then you came back. So what did you do next? Did you do a postdoc?
Yeah, so then I went back to the US. I went to Yale, to New Haven, Connecticut, to Ira Mellman.
Oh yeah, sure.
Because I wanted to learn more cell biology. So he has also been instrumental. I think all the people I've met and had the fortune to work with have really, I feel very blessed. But he's a fantastic person and cell biologist, and I wanted to learn more about antigen processing and presentation and how viruses can both sort of actively sometimes influence that process, but it's also a very important part of how we fight viral infections, of course. So I joined his lab then quite soon after I've been there for a year or so. Then he told us that he was moving to California. So the better part of the lab moved with him. Right. So then I was in San Francisco for three additional years at Genentech.
He moved to a company, right? Genentech, yeah. So
that was also, as it turned out, a great experience that I didn't anticipate at all to have as a postdoc. But it's
an amazing company.
Yeah, It really is a great city and fantastic people. It's just very far from Sweden. That's, I think, the only drawback for me. Otherwise, I would have loved to stay. But my husband was here in Sweden. So after
4 years?
For 5.
5 years?
5 years, actually. So then I got, an opportunity to start my own lab here.
Right.
And I took it. And I'm very happy about that, too.
Sure.
Research is nice because you can continue to work together even if you are far apart.
It's good to have different experiences, though, especially to go other countries and see what science is like and have that influence you and so forth. And you've been to some very good places, right?
Yeah.
As the NIH, Yale, Genentech, that's great. Do you remember when you wanted to do science? How far back that came?
No, I also had a very mixed I was interested in almost everything growing up, sports and music and literature and science. And my parents are physicians, both of them. And for a while it was the only thing I definitely didn't want to be. And in the end I didn't go to medical school. I did a master's program in mathematics and biology. I'm very happy with that. But then I think sort of quite early on there, it was clear that I wanted to try to pursue a research career. And immunology and infectious diseases was, I think I was also very influenced by the lecturers that I happened to come across. That they were the ones I had in immunology were very inspirational.
Yeah. Okay, so 3 very different stories, but all end up in the same place and all doing science. So let's talk about some science. Jan, I want to start with you. Okay. Now, I know you work on mostly HIV, but you also work on enterovirus D68, which we started to work on 4 years ago. I spent my career working on poliovirus.
So that's close, at least.
So, as you know, it's about to be eradicated globally, although it's been eradicated in Sweden for many years, actually. It's quite interesting.
Yeah.
Erling Norby told me last night it was eradicated using the inactivated vaccine, which Albert Sabin, who made the infectious vaccine, said could not be possible. You can't do it with just the inactivated, but it worked here. In
Sweden. We're one of the few countries that use the inactivated polio vaccine, and it's been very successful. But it requires, of course, a stable healthcare system where you can repeat vaccinations, because it's more challenging in that way.
Yeah, so whether it would have worked in the US, we don't know, because we started with inactivated vaccine in 1955 and we stopped in 1961. But if we had kept using it, who knows? No. We might have been able to eradicate it, but we'll never know the answer. Unfortunately, using the OPV of Sabin, you get vaccine-associated polio, which is highly unfortunate.
Yeah, that's sort of one of the things that we've been able to avoid here.
Yeah. So we searched for other viruses, and enterovirus, as you know, all of a sudden there was an epidemic in 2014.
Yeah.
And did that happen here in Sweden as well?
Yeah, it did. It affected all of at least Europe, and I think we had virtually the same sort of scale of problem as you had in the US and in Canada, and it also affected other parts of the world, maybe less studied.
And so since then, there's been an outbreak in 2016 and 2018, and we're expecting one next year. And I don't know about here, but in the US, there's a lot of activity. There are consortia being formed to try and
it's the same, both in Sweden and in Europe. So we got involved in part because we are a reference lab and the biggest clinical lab in Sweden. So I'm partly sort of in research, partly in clinical microbiology. But then there are many sort of things that we have learned from HIV that we can use on other RNA viruses. So it's relatively easy to transition. So it was already started in a way. So we started working on enterovirus when this first outbreak was reported. So we checked whether we had cases in Sweden. We had some that were, Sweden is a small country, so the numbers are much lower. So we only have seen a few patients with this paralysis, but many with severe or mild respiratory infection. And we had a really huge outbreak in 2016 and fewer in 2018 than many other countries. Maybe because we had so much in 2016.
I mentioned this morning, the population of Sweden is like 10 million.
Yeah.
Which is less than New York City. I'm just still amazed. The place I work has more people than this whole country, yet this country is very big.
It is big, yeah. It's more similar to Canada when it comes to sort of density of population, and many parts are hardly inhabited at all, and then there are some centers like Stockholm has almost 25%, or the Stockholm area has 25% of the entire population.
Right.
20%.
Someone said this morning that you have everyone's number, so you can do great epidemiology, right?
Absolutely, and that's sort of what I was alluding to when I talked about the possibility to use an inactivated polio vaccine. That we really have good track of everyone, for good and bad.
So let me ask you a few things about this Entero 68. So in the U.S. there continues to be some puzzlement. First of all, the virus was known since '62.
Yeah.
So why all of a sudden is there an outbreak in 2014? Do you have any ideas about that?
Well, of course we've thought about it, as I'm sure that you have and many others who have worked on this new or not new virus.
Yeah.
Because there are seroepidemiological studies, and maybe, I mean, I thought about maybe we could collaborate on that.
Yeah.
From Finland, for instance, showing that almost everyone who is adult has antibodies to this virus. So it's been around, but these more severe disease symptoms have not been recognized or have not been there. And I think it's a little of both. But there is also this discussion about whether the strains that are circulating now are more neurovirulent or not. And I know that you have worked on that. I haven't worked directly on that, but it's really an interesting topic, whether the virus has changed in its in the way it interacts with us, and the way whether it sort of can invade the central nervous system more easily now than earlier.
Yeah, we find that all the strains from 62 to the present are capable of infecting nerve cells. Whether the virus can get there is a different question, and we don't. That's neuroinvasion, and we don't know anything about that.
Yeah, there was one recent paper suggesting that transport through the nerves from the periphery to the central nervous system might be better for the newer strains. But I mean, there are conflicting data on this, and there are quite few studies still, and they are in part in sort of animal systems and things like that. So
I think we definitely need better animal models and to look at these questions. We just don't have them yet. So I mean, the idea that it's been around already is interesting, but there's a peak of AFM, acute flaccid myelitis, in 2014, and we were always looking at it. I mean, I think after that year, the U.S. started to do surveillance really for AFM, but previously there was always a low level throughout the world. So I think we would have seen peaks like that. So I really don't understand that. Do you understand why it's every 2 years?
Well, I think that's sort of similar or reminiscent of many other viruses, like respiratory syncytial virus (RSV), which causes severe respiratory disease in really young children, which tends to be every second year. And so, and there are both studies and sort of epidemiologically data suggesting that there has to be enough of susceptibles. So in 1 year there are too few newborns. So maybe every other year, and it may depend on population density and stuff like that. So these cycles may differ in less or more populated areas.
Also, with other enteroviruses, we know that population immunity can control that as well. You get an outbreak, you get a lot of people immunized, and then it takes 2 years to get sufficient numbers.
So my research, part of my research has been to look at the molecular epidemiology and evolution of the virus, trying to understand these outbreaks and whether there is whether the variants that make out sort of the change from 2014 to 2016 is due to changes in areas of the virus which are important for immunity, neutralizing epitopes. We have some molecular indications, but we haven't formally tested by neutralization assays, sort of tests on live virus.
So you just published a paper on evolution of 68 by whole genome deep sequencing.
Yeah.
So tell us how you do that and what you learned from it.
Well, it's a method that's becoming sort of has become available for many different pathogens, and also for us studying us humans, because sequencing technology has improved or changed dramatically in the last few years. So it's possible to do much more sort of in-depth DNA analysis. So we make use of that, and we sequence the entire virus instead of just small bits.
It's from a swab, right?
Yeah, from a nasal swab, and we do it, and then we use these technologies called next-gen sequencing, and because the throughput for these analyses is huge. We can sequence many samples at the same time. We can sequence the entire genome of the virus, which is still very small, and we can sequence also the different variants that exist at the same time in a sample. And then we, together with biophysicists in Basel, Richard Nee here, we do quite sophisticated bioinformatics and analysis of evolution and things like that.
So when you take the swab, you sequence the total RNA from it? Is that how, or do you?
No, so far we've done directed amplification and for the studies that are published, we've done it. We've amplified by PCR, which is a technique that is used in all of medicine basically. So we amplify the virus genome, which is 8,000 bases in 4 overlapping fragments, because it's easier to do this in smaller fragments. And then we just apply these sequencing technologies. Okay. But we are working on more general methods, and in part, this is something that we do for our enterovirus surveillance, because we want a generic method that we can use for any enterovirus or rhinovirus for that matter, polio as well. So that's in the pipeline. We have ideas on how to do it in a more generic way, and there are different approaches there. Some that are entirely generic, so we could sequence any RNA virus, and some that are still directed towards enteroviruses.
So CDC says if you do the total sequence of a nasal wash, you get so many viruses that you can't tell which one is actually associated with.
That's a challenge, and this is I'm responsible for development in our diagnostic unit in the clinical microbiology at the hospital, and we are working on implementing metagenomic diagnostics, which is just blind sequencing, large scale, and then search. So which pathogens do we have in this sample? And as you say, one of the main challenges, apart from having sort of good sensitivity, is to sort out which of all of the pathogens that we find are the relevant ones. Yeah. But that's in part, we already have that problem to some extent, and you might see quite often that when you do the usual panel on a child with respiratory infections, you have several viruses detected, and certainly one of them is probably causing the disease. And just because you don't look for more, these other pathogens are still there.
Well, CDC says when we find 68 in the CSF, then we'll believe that it's associated with paralysis. And that's been found very rarely.
Yeah, it's strange, and that's another interesting thing about this disease, that even though the symptoms, at least in the ones that have neurological disease, the symptoms are neurological. The virus is rarely found in the CSF or in the central nervous system, but much more easily in nasal swabs.
So, in your samples that you have sequenced, are they from children with a respiratory infection?
Most of them, but a few are from patients, both children and a few adults with paralysis.
Paralysis, okay. Yeah. But no CSF isolates, right?
No.
Yeah. Yeah, that's a...
We haven't even tried isolation. So, of course, we could reconstruct a virus from the amplified material, but we haven't done that.
That would actually be very interesting, because especially from the kids, the child and the adult with AFM, acute flaccid myelitis, to reconstruct the virus and look at its properties.
Yeah, absolutely, and that's something that I mean, it's relatively easy, but everything it takes time.
Of course,
and resources.
Now, when you look at the sequences that you have assembled in this paper, what do you learn from that?
Well, we've learned that the outbreaks into 2014 and 2016 were dominated by a sort of a clone, almost like almost all viruses in different patients was very similar. We have indications of we also look at sort of genetic variation within a patient sample, and we can see signs of immune pressure on epitopes that are known to be neutralizing. So that could be something that could drive evolution of the virus. We've seen by deep sequencing that some patients have dual infections, which hasn't been reported before, and which is both sort of expected and unexpected, because we had really massive outbreaks. There was 1 week in the fall of 2016 where we had 25% of all our samples that came in for respiratory testing that were positive for enterovirus D68.
Wow.
And then you can imagine that some have been exposed twice, and haven't been had time to mount immunity.
Yeah.
But an interesting thing that we see now, because we also have tested not only Swedish samples, but from across Europe in 2018. This is not published yet, but we see that this year, even though there was quite a lot of enterovirus D68 in circulation, maybe less in Sweden than in some other places, there is not a dominant strain. The virus is all over the place, and so it seems like it hasn't decided where to go next. And it will be really interesting to see if that pattern holds, but I would expect that when we have a larger outbreak, it will be dominated again by a single strain.
So you think the pattern is that there's an imported strain? And that's,
well, we see clear evidence by molecular epidemiology that the viruses that we detected in Stockholm and to some extent in other parts of Sweden in 2016 especially, because we had a lot of samples, they were mixed with international samples. So there was the virus was imported several times and then spread locally. And that's the same pattern that has been described for influenza and also other respiratory infections.
So I wanted to ask you, in these individuals where you see multiple infections, you see any evidence for recombination? Because enteroviruses do that, right?
Well,
just like HIV, I guess. Right?
It's an interesting question, and it's because of the technology we use when we just chop all of the DNA into small bits. It's difficult to say which bits belong to which ones from the original virus population. So by using newer technology, and that's something that we're looking at right now. Nanopore is an interesting technology where you can sequence
longer, right?
Yeah, the whole virus.
Do a longer read, but it's got more errors. But
but for recombination, it would be good enough. So that's that will be interesting to see whether we have recombination in these cases with double infection.
Right. And one last thing two last things I want to ask you. First, do you have a website where you put the sequence data, nextstrain.org, right?
Yeah.
So that's very interesting. What is that exactly?
It's my collaborator in Basel, Richard here, who together with other people have created this open source or freely available resource for real-time analysis of molecular epidemiology and phylogenetic of different viruses. They started out with influenza. So it was called Next Flu, and one of the ambitions was to predict which would be the next flu. So they also worked together with the CDC and the European CDC in trying to help in choosing the vaccine strains for the next year for influenza. And then they expanded to other pathogens. I suggest, because we have collaborated on HIV, and when we started work on enterovirus D68, I suggested that they would add D68 to the Next Strain website. So I inspired them to do that, and then we do the sequencing, and we also collect, of course, other published sequences and put it freely available. And also the tool is quite nice. It's really interactive.
So you can go and see the phylogenetic trees of all the sequences, right?
You can look at different parts of the genome. You can check specific mutations, how they track through time, and things like that.
You can also animate the spread of viruses, right, globally.
Yeah.
It's quite nice. Nextstrain.org, right?
Yeah.
Okay. The other thing I wanted to ask you, and this is actually might be of interest to Anna as well. So you're interested in infections of the lung, right? So this virus infects the respiratory tract, upper and lower. There's no viremia, yet in a small fraction of kids it gets into the CNS somehow. We think so, right? How does it get in? Do you have any thoughts?
There are indications that it's similar to poliovirus, which is also an enterovirus, and some other enteroviruses, that the virus is transported from the periphery to the central nervous system through the nerves.
Right.
So that's also rabies does the same thing, and herpes viruses, at least some herpes simplex and varicella zoster virus does the same thing. So this is an established way for viruses to transport itself from the periphery to the central nervous system, and some do it quite well, or almost always, like herpes virus, and some do it maybe more by chance and mistake, like polio and enterovirus D68. So that's my guess. And yours?
Well, I mean, since there's no viremia, I don't think the virus can get peripheral. It would have to be from the lung. So that's possible, but people have told me that the innervation of the lung is not the same place in the brain where you're seeing the lesions.
Okay, yeah.
So that's probably not true. The other thing, maybe I wanted to ask you, if this virus infected DCs or monocytes, could they traffic into the CNS, or is that completely unlikely and bring the virus there?
Yeah, that's a good question. I just know that there are definitely studies asking similar questions with toxoplasma, for example. I know Antonio Barragan at Stockholm University has done that, where you can see that that's how it gets into the central nervous system via infecting dendritic cells that then migrate. So I assume it could happen.
Maybe. So
it's a general trend sort of in immunology that we are starting to sort of revise this idea that the blood-brain barrier is a real barrier, and that, for example, in the choroid plexus is believed to be one way in for specific immune cells. And so I think that's plausible, but in this particular case, I'm not aware of.
And the third possibility is just a paper published a few weeks ago from Roberto Cattaneo, showing that one of the measles receptors can actually mediate the transfer of cytoplasmic contents from cell to cell. And he's shown that measles virus can go from cell to cell this way. So it's possible that if a enteral-68 infected cell got into the CNS somehow, that the virus could go from cell to cell without having to have extracellular virus. So and because the polio receptor, at least, is the same family as one of the measles receptors. It's kind of attractive. I don't know what the entero-68 receptor is yet. There are a lot of candidates out there, but it's not clear which is the receptor or a receptor. But it's an interesting question. Anyway, you might be interested in this virus. You should talk because you're into the lung, and maybe you could do something. I know you teach virology, right?
Yeah,
so
I do.
I want to come back after we talk about some other science here. If we have some time, I would like to talk, because I love to teach virology.
Yeah, me
I'd love
too.
to talk about it. So Petter, I wanted to start by asking you to tell us about human systems immunology.
What it is?
Yeah, so I have to tell you a joke.
Yeah, sure.
I saw something.
Sidney Brenner?
What's that?
Was it Sidney Brenner?
Oh, he tells a lot of jokes. He used to.
He used to, yeah.
He might have said this. He said that systems biology is physiology with marketing. I don't know if he said that or not. Is that the same with humans?
No, Sidney Brenner said that systems biology is low input, high throughput, no output biology.
That's good. I hadn't heard that one.
Genius, yeah.
You probably don't agree with that.
No, I do. I mean, I do that. We have to define what we mean, right? And so, systems immunology, just like systems biology, is one of these buzz terms, right? And it can mean absolutely nothing. So what I mean when I talk about systems immunology is sort of shifting the focus from studying immunology one cell type at a time, which is what we've traditionally done, because the system is really complicated. And so we have to sort of dig deep to understand individual cells and so on. But to try to understand how the system itself is wired at a more global level, we need to take a step back and start looking at how the different cells and the proteins that they produce interact with each other to determine an immune response, for example. Because an immune response is never carried out by an individual cell type. It's always a coordinated effort by antigen-presenting cells and effector cells and so on. And to try to understand how that complicated machinery starts moving, me and many other immunologists have started to try to look at the system at this more global level. I mean, it's never going to be a fully global picture because we can't take all the cells in all the tissues into account. But in my group, we're trying to use blood samples in the smartest way that we think we can to try to learn as much as possible from looking at many of the components of the immune system in that particular blood sample. And I think it's sort of it's a complementary approach to studying the immune system. And that's what I mean by human systems immunology. But other people mean other things.
Now, so I read a thesis of one of your students.
Right.
That Kim sent to me. And so he said that he wrote the same thing you've said, that traditional approaches only look at a cell or a few cells. But that's all we could do
at the time,
10 years ago.
Sure.
So there's nothing wrong with that, and we've learned a lot, right?
Yeah.
So I think a part of the criticism of systems immunology is that everyone says, oh, the old stuff is no good anymore.
Yeah,
right? And that's bound to raise people's
absolutely.
hackles.
But isn't that's always the thing in science? We're sort of following trends all the time, right? And right now it's sort of a trend to look at, gather lots of data. It's a pretty clear trend in biology as a whole, right? But that doesn't mean that we sometimes have to dig deep into specific mechanisms. But I think for me, at least in my team, we try to go back and forth, and we look at some global patterns, and then we try to look at the details of those. What determines a specific cell's reaction? What's the what's the transcriptional program? What are the regulatory mechanisms? Right? That might be an in-depth analysis. Yeah. And so I think they're complementary approaches that, but it is enabled by new technology.
So tell us what are some of the new technologies that enable systems immunology?
So first and foremost, the technologies must work at the single cell level for this to be possible, because we need to be able to look at individual cells and take sort of the heterogeneity of cells into account. And there's been a pretty dramatic development in the last 5 years or so of different methods that allow increasingly complicated measurements to be made on single cells. So whether it's a transcriptome, so mRNA molecules, whether it's DNA, proteins, epigenetic marks and traits. All of these different things are now, and actually now the sort of the latest development has been to put these different things together in the same cell. So to measure both protein expression and mRNA, and epigenetics, for example, from the same single cell. And this is sort of where the field is right now. And it's been enabled by next-gen sequencing type methods that Jan already mentioned. It's been another sort of development which we work a lot with has been to do more protein measurement from individual cells. And that is based, for example, mass cytometry is a method that we use a lot, which is sort of a next generation cytometry. Traditional methods have been based on fluorescence-based cytometry, which is really a workhorse in immunology and has been for for several decades. And still is, I would say. But the fluorescence-based methods are limited in the number of parameters that we can measure on the single cell because of spectral overlap and so on. So mass cytometry has been developed to increase that number. So now we're measuring something close to 50 parameters in individual cells. And doing that for millions of cells gives you a very rich data set from a small blood sample.
This can be intracellular as well as surface proteins.
Right. You can permeabilize the membranes of the cells and get antibodies to stain intracellular proteins. Okay. You can combine mRNA with protein measurements and so on.
So all these data, how can you manage it?
Right. So that is the challenge, right? And that's I think that's also one of the reasons why there's been a lot of criticism, is that you gather a lot of data. And sometimes investigators present the data without any real conclusion in terms of new biology. It's just a lot of data. And I think a lot of the criticism comes from the fact that we have generated a lot of data, but we've failed to extract new biology. And I think that's what Sidney Brenner is alluding to in his comment. And I think that's correct. We need to think about what's the hypothesis, what are we trying to learn, and then what is the data actually telling us when we try to distill the large amount of data into something meaningful. It's a real challenge, especially when we're trying to integrate different kinds of data, but it also has a lot of potential. And so I think yeah.
So obviously a key part is computational approaches, right? Software and hardware, which you could have all these data collections, but if you didn't have a computer to analyze it, you'd be lost, right?
Sure. And obviously...
and someone to program the computer.
Absolutely.
Yes.
No.
Yes, that's right.
You're absolutely right. Those are essential parts of modern research in this field, for sure.
You have to have a way of displaying the data. And I mean, to a certain extent, the computer can look for patterns, but there's nothing like the human eye to look for pattern, right? I just saw that there's this wonderful website where you can display the temperature of your town every year in colors.
Oh, yeah.
And if you look for past 100 years, you could see blue, 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 red. And that's the kind of pattern you understand. So you have to do the same thing for your data. Can you do that? Are you getting
you can. And but you also need to be sort of an informed interpreter. You need to know what you're looking for. You need to have some background information. If you just try to, throw algorithms to try to tell you, you're going to get lost. And so what we typically do is we would generate the data, we will make sure it's good quality, and then we start asking simple questions. In our particular case, what things are changing over time in a newborn child and its immune system? What are the differences between a boy or a girl? And how, simple things like that. And then see what we can learn from these patterns.
I don't know if you know this, probably not, but one of your papers we discussed on TWiV a while ago, this one where you looked at the antiviral antibodies in newborns, right?
Mm-hmm. Yeah.
Over time. And this is an example of systems immunology.
I think so, yeah.
Because you use a a chip-based array to look for antibodies, right?
It's a phage display.
Phage display, right.
So it's a sequencing-based readout.
So the antibodies bind phages and then you sequence, yeah. But you would say it's systems immunology.
Right. I mean, it's a global, to some extent at least, global analysis of the antiviral antibodies, IgG antibodies, I should say.
You look specifically at IgG, right? So in that study, so there were a couple of things that we really liked. First of all, the most common viruses, mostly herpes viruses and rhinoviruses, that kids have antibodies to, and they're getting that from their moms, right?
Right. So we think the antibodies we're looking at are maternal because we're looking during the first weeks of life when the IgG antibodies, at least according to previous literature, the children are not producing their own IgGs. And some reports say up to 15 weeks of life.
And IgM don't come across the
IgM
placenta,
doesn't cross the placenta. So that's why the IgGs before 15 weeks would be maternal. Whether that is exactly right or not, we don't know. But at least in the time window we're looking, we think most of the IgGs are maternal.
And maybe the most surprising things is that the preterm babies have similar IgG.
Surprising to us too, yeah. We expected a very different repertoire because as clinicians, as pediatricians, we learn that preterms are born before they get the maternal antibodies, largely at least. And therefore they are susceptible to infections. And that's why we need to be particularly careful with the preterm babies and give them passive immunity for RSV, for example, and other things. But, when we look sort of in an unbiased, I want to say an unbiased way, just looking at the serum samples from preterms and terms, we don't really see a difference between the repertoire of IgG from the mother.
So why are they more susceptible? Is it because the immune system is less developed? You need more than antibodies?
I think like this. I think there are a couple of things to say here. Number one, are they more susceptible? Do we know that?
More fragile, maybe.
Right. And so you could imagine that what is susceptibility? Is it to be more exposed, right? Or once exposed, you get more sick, right? And the second is probably true, that the preterms get more severely ill when infected. And that could be either because their immune system is unable to fight the virus, or it could be because they are just, their lungs are sick for other reasons. So if you have in pneumonia, when you have bronchopulmonary dysplasia, for example, you get very, very ill. Your reserve capacity is very low. And I think, another thing I always like to remind people of is that most of the preterms, at least the very preterm and extremely preterm, they're cared for in an intensive care unit. And if you take adults and you put them in an intensive care unit, they're also more susceptible to infections than adults not in an intensive care unit. And so I think we really need to think about this. But there are also reports, historical reports, that have suggested that the immune system of preterm is less capable, less evolved. I think the bets are still out there. We're looking at it actively. At least the maternal antibody don't seem to be the main factor, according to our data.
So what else can you do with this? I mean, going forward, are you interested in understanding this question? And
absolutely. We're working very actively at that. We're trying to look at the functional development of the immune system in preterms and terms. We're also trying to distinguish very ill preterms from not so ill preterms and how their immune systems are affected. We're looking at the newborns and how the immune system evolves in relation to the metagenome, because as in the gut specifically, we're looking at fecal samples and doing this kind of shotgun sequencing that you mentioned to try to understand, the babies are colonized by bacteria and viruses and fungi and parasites and everything. And the immune system needs to sort of evolve in parallel or in relation to that colonizing metagenome, if you will. How does that happen? How does that influence infectious disease susceptibility and other things? So it's a very complex process, but that's what we're mainly focusing on at the moment in my lab.
So I understand that this colonization mainly happens shortly after birth, but someone told me that as babies are drinking amniotic fluid, that has phages in it, and that can modulate the microbiome on birth.
It's an interesting
new
development. Yeah,
what you get from the mother could make a difference.
It's a very contentious issue in general, whether the womb is sterile or not, right? And there was a pretty recent study from the UK, which was very interesting in my opinion, showing pretty convincingly, I think, that the bacterial colonization seems to happen after birth.
Right.
That doesn't mean that the immune cells prior to birth cannot be seeing parts of bacteria and viruses and so on from that circulate in the maternal circulation, right? So it's and in support of that, there is quite a lot of evidence now suggesting that you have antigen-experienced immune cells in fetuses, suggesting that they are seeing something. Whether it's live bugs, we don't know, I would say. But anyway, so I think all of these things probably shape the immune system. But what we do know is that after birth, there's sort of an explosion of exposures that the baby is seeing, of all kinds of things, as you can imagine. And that definitely has a pretty significant impact on the immune system of the newborn.
Now, this study on the antiviral,
right?
Was this done with Swedish babies?
Right. It was a cohort that we are following here in Stockholm. We're actively trying to also do additional birth cohorts in other parts of the world, where you can imagine exposures to be very different, both prenatally and postnatally, and so that's a very active sort of development we're pushing. But the study was in Sweden, yes.
Yeah, I wanted to ask you about other countries, because I've learned on my trip, this is my first trip, that everything is perfect in Sweden. Everything works and it's clean and ordered. So you can imagine that the babies at birth are...
We're happy that you got that impression.
It's not true.
I think every country or place has its own sort of benefits and drawbacks, and that comes to Sweden as well. Some things are very well organized, but some could be still better.
Well, from what I've seen so far, I would live here, except that it's too cold probably in the winter and too dark.
Yeah.
I'm not sure.
I agree, yeah. I still live here, though.
But that's what I've been told, because I don't know. I haven't lived here.
But it's not colder than many sort of inland US or Canadian cities. Sure. Stockholm is not very cold, but it's dark.
Yeah, well, I was told it gets dark at 3 p.m. in the winter. That's a bit much.
Yeah, it is.
Well, you just stay inside and do mass cytometry.
Well, that's when you go and do your studies abroad, right?
That's right. I wanted to ask you one more thing, Petter. There's a BioArchive manuscript, which is fascinating, about self-monitoring of blood, systems-level immunomonitoring using self-sampled capillary blood. Mm. Tell us about that.
Yeah, we're working on that. It's
is that submitted, by the way? Because it's in BioArchive.
Yeah, it's under review. So that is a interesting development. So we've worked. I told you earlier that we've been trying to get as much data as we possibly can from blood, and given our studies in preterm babies, we were forced to miniaturize the amount of blood that we can analyze, because the very sick, very small preterm babies, born in week 22, 23, are very susceptible, and we can't take a lot of blood for research from them. And so we had to miniaturize. So now we're down to about 100 microliter of total blood starting volume to do all of these analyses that we want to do. And so that was enabled by the fact that we're able to preserve whole blood directly in the clinic. When the blood is drawn, we're able to preserve it using a preservative solution that can be frozen. And then the cells can be thawed and analyzed at some later time point. So that's one important development. The other thing is that now that we can use small volumes, such as 100 microliters, you can start imagining capillary blood, where you can draw those kinds of volumes and you don't have to do any puncture. So that was another part of the development. And then thirdly, there are these different systems for profile or sampling capillary blood. We're using microneedles that penetrate into the skin, but they don't go deep enough to cause pain. So by using such systems that typically also involve vacuum, so that you get or capillary forces that draw the blood out, you can, in a very pain-free manner, by just placing a device with 100 microneedles, you place it on the skin, and then you activate it, and then you can draw out 100 microliters of blood. And so this is something that we've been working very actively with, and in collaboration with partners in the US and others. And so, I foresee a future in which we'll be able to basically send these devices home to people, and they can sample themselves, put the device in the freezer, and then we go and collect it. That would allow people to sample themselves. You can sample someone 10 times in a day because it's pain-free and you don't draw a lot of blood. You can imagine doing this in children where the barrier for blood sampling is always a lot higher. So we're pretty excited about this. I think this will open up new types of studies. Also remote studies where you don't have clinical staff or people who can preserve the samples are enabled by this kind of approach. So
I'm used to going and giving 10 tubes of blood.
Yeah, right. Yeah.
And this, but so
what it's an important sort of thing that also for other types of samples, that we tend to get samples from the most sick, or so that is the spectrum of samples and pathogens and immune reactions that we see are not really representative. So by having the possibility to sample in an easy way, also from people at home, that opens up many possibilities. And the same goes for sort of easier ways of collecting swabs and things like that.
I think in general there's a lot more we can do with samples. We sort of, this 10 tubes, it's really such a waste, It's
a waste.
Really
they throw most of it away, right?
I mean, you do. You prepare your PBMCs and then you throw away them. That's another point, is that we're finding that when you prepare PBMCs, obviously you're throwing your neutrophils out, and then you're no longer looking at a population that is representative of the entire blood. And so by moving towards whole blood analysis, we're seeing a lot less technical variation and clearer biological signals. So that's another part of the development, to try to go closer towards the sort of in vivo situation.
We had a similar thing for one of the enterovirus D68 studies, where we wanted to do HLA typing, and we didn't have any blood samples, and we didn't have really the patients to draw samples. So we tried doing HLA typing on the nasopharyngeal samples, which is just minute, and it was
fine.
Fine, easy.
You get cells from the epithelial and nothing,
So you, in 100 microliters, you can do flow cytometry,
or this mass cytometry.
Cytometry as well.
Yeah. So we get sufficient cell counts for that, because if you're not throwing away everything and you're processing and you're actually using the entire volume, you're preserving it, freezing it, and
it's in a preservative, so the cells are not lysing, right?
They're not lysing. They're not alive though. They're fixed, but they're not. They're intact in their membrane structure.
Could you do antibody surveys with this?
Absolutely. That's what the whole antibody study that you mentioned earlier was done using these 100 microliter. Because for serology, you need almost nothing, right? It's very, you need very low volumes, typically, of plasma or serum for that.
Can you do blood chemistry, liver enzymes, you could do anything?
Yeah, absolutely.
So basically anything that you would do in a clinical lab?
Yeah, blood chemistry is interesting because the clinical labs typically want quite a lot.
Yeah.
We typically send, half a milliliter from the clinical blood draws from children to do clinical chemistry in the standard lab, which is really, you know, there must be room for improvement there in terms of getting down
I think
to
the
low.
machines that then does the automated analysis uses like a fraction.
I'm sure, but it's backup, right?
Yeah.
And so I think.
And it's also the sort of the sample collection systems that we're used to.
Right. But I think, you know, now that, especially with susceptible or fragile preterms, we're learning that, you know, sometimes during the first months of life, we might have drawn 90% of their blood volume and we have to replenish it with adult donor blood. I mean, that's not physiological, right?
Right.
You take away all the stem cells, you're giving back erythrocyte concentrate. It's it cannot be beneficial, to say that, at least. You know, so I think you know, this is something I can talk for a long time about. But
okay, it's great. I love it. Yeah, 100 microliters. All right, let's move on to Anna. So you're interested in the lungs and infections, right?
Right.
And different cell types in the lungs. So tell us how you study that.
Right. So I think it's an interesting continuation in a way to the stuff that you've talked about, because while on one hand, I think we can definitely improve sample collection from blood, and we have already, I would say most of what we know about human immunology comes from circulating immune cells, because it's the most accessible sample we can get. But we still, I would say, and many, I think, with us argue that it's probably not always sufficient to only look in blood to understand the whole picture.
I agree.
Especially when you study pathogens or diseases that are quite localized to a certain place. So in the lab right now, we are very focused on respiratory viral infections, so mainly influenza, also hantavirus infection, and then different pulmonary inflammatory disorders. And when we compare immune cell, both composition but also function, from the same patients, that we retrieve samples from the respiratory system, either via nasopharyngeal aspirates that we do on flu patients, or bronchoscopies that we do on other patient groups. And we compare them with blood samples from the same patient at the same time. They are quite different.
Oh, I'm sure.
So that is something that we are quite interested in to understand, sort of, especially with, so we are mainly focusing on monocytes and dendritic cells, and they, by sort of virtue of their biology, also migrate a lot, even under steady state conditions. So they are sort of there to sample their environment, to look for foreign things, and if they sense something, a dendritic cell would then go to a lymph node to present it to a T cell. But is this shifted or altered during disease, and does it matter what composition of these immune cells you have at the site of infection, for example, during an acute viral infection?
So you sample the respiratory tract at multiple levels, right?
Right.
I have a picture from one of your papers. There are four levels, right?
Yeah. So that may be, so when we really started to embark on this, we did studies on healthy controls, because we realized that we didn't really have a lot of detailed data to compare with. So that was something we spent a lot of time on in the beginning. So we looked at different sort of parts of the lungs, and also took small endobronchial biopsies to look at the cells that, because of course when you lavage the lung and retrieve immune cells or cells, what is that a reflection of compared to the cells that are still in the tissue? Because it's most likely those cells that are, still around to sort of deal with an infection. Or
when you wash, you get the surface, the cells that are sitting on the epithelial surface, right?
Yeah.
But are those DCs and monocytes? Are they moving around on the surface? Or?
So that is also, I would say, quite poorly studied in humans for sure, because it's very difficult to do that. But there have been some studies on pigs trying to look at to what extent immune cells in general just go in and out, sort of that they cross the alveolar space, and they do to some. So it's not just sort of a dead end.
They go in. They in and out. Oh yeah, because they have to go back with sampled antigens.
Yeah,
right? And they're doing this constitutively, I guess, to sample the lumen of the respiratory tract. Right?
Yeah, that's the
idea.
The idea, at least.
It's hard to, because you have to have a volunteer who allows you to take biopsies, right, of their lung and so forth. I was talking with someone a surgeon who, he said he could go in and not get the same place every time so you don't damage. Because if you take a biopsy, you're going to get inflammation there, right? So the next time you go, you want to go somewhere else. And he was saying how he was better than the residents doing this. Anyway, so you take these cells out from different levels. And one of the things is can you develop better sampling methods? But right. I don't know, it's as hard to get at, so I'm not sure you could.
Of course, to access the lung is a challenge, and in some, circumstances, it's, at least with the methods we have right now, it's not feasible. But I'm sure, these things can improve as well if someone sort of decides to devote their life to it. But for certain patient groups, it's part of the procedure of obtaining a diagnosis, and then it's relatively easy for us to also get research samples. For influenza patients, for example, we would typically then not have them undergo bronchoscopy. But then we try to sample the upper part of the sort of the nasopharynx to get an idea of what it looks like at the site of infection. And
so that's easier to do.
It's easier to do, although it's also required quite a lot of sort of methods development, I would say, because we also then want to get enough sample to do flow cytometry and quite careful sort of immunological characterization of these cells. But that is, a lot of that work is done by Sindhu, who is listening in here.
So one of the things you do is to take out these cells and infect them, right? In culture.
Yeah, so
you add virus. I would say you add virus to them, right?
Yes, we do that too. But I would say the lab as a whole, we sort of go from, we try to look in patients and see, how do the immune cells sort of behave if you have an acute influenza virus infection compared to when you're convalescent or compared to a healthy control. And then the idea, we can also do in vitro infections, but we typically wouldn't do that on the cells from the infected patient, but rather to try to understand phenomena that we observe in the patients, to understand them, and I would say go back and dissect this more. Then we do take a more reductionistic approach and add one thing at a time to see what is what.
When you take cells from an infected patient, though, you don't get multiple time points. You get one probably, right?
Yeah, so typically we would get an acute and then a convalescent sample. But that is something, there are studies where they've sampled influenza patients, for example, several times during the acute phase. But that also becomes, it's logistically a challenge to some extent, but also, especially when you want to sample the site of infection, there's a limit, I think, to how often you can do it without, then a blood sample is easier, for sure.
If you have an intubated patient in a ventilator, then
that's a different thing, yeah. But then you are also talking, and that is also something that we have discussed a lot, that many of the studies are on the most severely ill, and partly maybe for that reason. Whereas if you want to go for sort of questions like why does the same virus cause such a spread of disease severity from not sick at all to death? You need also to try to sample the much less severely ill. And here it's something, I mean, we should definitely talk. Because, no, but I mean, we are still limited to, even if we try decisively to take a broader approach, we would typically include patients who still seek healthcare, which for influenza, we know, at least in Sweden, that most people who are infected, they probably sort of suck it up and they stay at home. They don't go to the hospital. But they would be very interesting to sample, to understand what they look like.
Do you compare young and middle and older people in this respect, taking these cells from their tract?
Yes, so far we have only included adult patients. There are also studies on children by others, and it would be very interesting to do that too. But I think it's also about sort of deciding what to do first to make sure that you get to the end of your study.
Yeah, well, you have to work with clinicians, right, to do this. So a lot of it's not in your control. It's harder, right, to do.
I mean, yes and no. I would say that for the flu patient study and also for our other sort of very patient-oriented studies, we work closely or have sort of established our own group of people. So typically for the flu study, which is also limited, for better or worse, but we have flu season, so it's only a certain part of the year when we can sample these patients. Then I usually have medical students that are in my lab that sit in the hospital and with the help of triage nurses and attending clinicians include suspected flu patients, because they are also a group of patients that would otherwise probably be asked to go home. So
you talked earlier about everything being perfect in Sweden, and we said it's not. But one of the benefits of being in Sweden, which is one of the main reasons why I'm here, is that it's easier, in my experience, to do studies, clinical studies of human patients, and also combining your sort of career as a researcher and a clinician than in many other places. It's much sort of a greater divide between healthcare and research in many other places than here.
Yes, I agree. So I'm listening to what you're doing, and I'm thinking I would never touch this.
No,
because it's
and
too
I would
hard
say it's true. It's one because I think been fortunate to be at very resourceful places in the US, but the type of studies that we run now in my lab would be difficult, not to say impossible to do, even when I had Genentech backing me up. I don't know if we could have done something like this.
So tell us what you find if you take a wash from an influenza infected patient and you look at DCs and monocytes. What are you finding?
So you find, maybe as you would expect, an infiltration of immune cells in general, but also of innate immune cells if you look quite early during the acute phase, which is, I guess, what we want to see. You also see neutrophil infiltration. And then what we are interested to understand is whether that is different. If we then follow the outcome of these patients, we are working under the hypothesis that what happens early on at the site of infection is probably what sets the stage for how this will develop. So that's what we're hoping to do in the long run. That will probably require, many years of work looking at both breadth of patients but also large numbers. But
and that hypothesis, would it be that the virus differs, the timing, or sort of, or is it human genetics, because even if you look very early and find differences, there could be different sort of underlying causes I'm not expecting you to answer, of course, but it's interesting to discuss.
And I think, again, you are right, of course, and we know already that all of these factors influence the development of the disease, both, the host genetics, the viral strain, and sort of pre-existing immunity. So it is most likely a combination. And this, I think, is also where you need to take a more global approach. It's difficult to study only one thing to answer a complex question. But I like the setup where you sort of, you go after something that, is a challenging question, and you look for a clinical phenomenon, and then you try to dissect that. And then I think you probably have to dissect it, even if you look at multiple factors, but we probably have to try to address them both jointly, but also one at a time. But I think it's very rewarding to work on something that we know is a problem that we would like to solve, rather than, sometimes at least, I've also, you could also have the other way around, that you work in a small animal model and you find something, and then you try to see if there is anything similar to this in humans or in real life. And of course, that's also a take on things, but I was always more intrigued by doing it the other way around.
So this idea that the early events determine the outcome of an infection, to look at that, you need to, so you're currently looking at acute and convalescent. Acute is too late. It's already,
right.
You want people where something happens early and there's no disease, right?
Yeah.
And see the difference. So for that, you need to probably infect people.
Right. So we have two, right. So you could do at least, maybe you can help us think of more ways, but one would be to use a sort of human infection model.
Right.
Those exist for sure. Then we're also trying to include household contacts, which are likely exposed but have yet to present with symptoms, and sample them. And then, so that we also do, but that's an even more challenging cohort to establish, but we do that.
The problem with the human challenge is you have to use only certain strains. And the real interest is to use a variety of strains which have different outcomes in people. And I think they use vaccine strains, basically. Right. And I don't think that's going to be very informative, because most people are not going to have a serious infection.
No, exactly. And then, of course, by definition, you would have a mild infection.
Right.
But on the other hand, maybe we have to do... often you also have to do what you can, or drop it altogether. And maybe if we combine what we can do, we can still, advance our understanding of this.
So let me understand this. So in these acutely infected patients, when you take a wash, you see an increase in the numbers of DCs and monocytes in the wash, right? And if you look in the blood at the same time, what do you see?
Then the frequencies tend to go down.
Do you think that's because they're migrating into the lung?
That's a very sort of tempting interpretation, at least. But that is also a challenge to really study in humans. There are some studies in humans labeling circulating monocytes, for example, and looking at their half-life. But of course, to then say that cell is the one that goes to a certain tissue is difficult. But we have seen it also in hantavirus infected patients. And there it was there the patients become viremic. So they have quite high viral loads in circulation. They're virtually devoid of dendritic cells in circulation when they peak in viral load. And at the same time, when you look in the lung, they are, their lungs are really filling up with immune cells, including dendritic cells.
And with flu, there's no viremia, right? So do you think the hantaviruses are actually lysing the cells in the blood, or?
Probably not. So we've studied that in vitro as well. So that was an example of where when we found this, we went back and did in vitro experiments to try to understand, is it that the cells, the virus can infect the cells and kill them. But hantaviruses are not really known to be very cytopathic.
Yeah.
So we see no evidence of that. Maybe we are also currently doing experiments. I mean, the primary target cell of hantavirus is endothelial cells.
Right.
So maybe you require contact, sort of immune cell endothelial contact between an infected endothelium, and that's what either alters adhesion or the leakage that is also associated with this infection. The increased.
Yeah. So what you said before is important, that you have to do what you can in people, right? But I also think a really good animal model helps a lot.
Definitely.
Because you can do large numbers. You will definitely get some animals who have less severe disease, and then you can see.
No,
right?
I agree.
So for flu, it's hard even with flu. The mouse models, the viruses that you use are mouse adapted, so they're not ideal. You're limited in the number of strains. The alternative is ferrets, which not great to work with.
Right, then it's
and they're
difficult
expensive.
to do immunology.
You can't do a lot in ferrets. It's hard. But maybe, so we're working with this mouse system called the collaborative cross, where you have basically outbred mice that more reflect the human genetic diversity. And you can find among this collection of a mouse that will respond to your virus, whereas none has ever before.
Right.
So it may be worth doing that and getting ones that can be infected with different influenza viruses, for example.
No, for sure. I mean, if I had the sort of resources and hands,
I understand,
think it would be. I would love to run a lab that spans all the way from sort of looking in patients, doing human immunology, and then go to mouse models, because I think all systems have their.
Sure.
Of course.
You can do different. You can ask different questions in different models, and we need probably all of them to understand what's going on.
Well,
there's also this really interesting development with respect to, from an immunologist's point of view, of wild mice or mice, the wildling mice. Have you heard about them? Recent publication from the NIH. So it's basically an embryo transfer from a normal B6 mouse to a wild mouse with a wild microbiome and all that. That gives you a lot more natural exposure, so to speak.
That's interesting.
And all of these different developments towards a more wild type mouse also has a lot of promise for immunology and potentially also from infectious disease type of experiments.
Now, that's what we're trying to do with the enterovirus. There's no good animal model, so we're going to try the collaborative cross to see if we can find a particular strain that will duplicate what happens in people, including the paralysis, maybe. The problem is these mice are $200 for a breeding pair.
Right.
And you need to look at least 20 different, so it's very expensive. So in that case, what you need to do is collaborate, like you said, I wish I could do everything. These days it's hard, so that you have to see collaborations. It's a place like this is good for that, so you can do that. Just a few more things. So I wanted to just chat briefly about teaching. Do you teach at all here?
Yeah, I don't teach as much as Jan. I know Jan is one of our most appreciated teachers in virology, but I help him a little bit with virology seminars sometimes, and I teach immunology to some extent.
And do it very good.
Oh, well.
Do you also teach some?
Not much. I mean, as a physician, we always teach younger colleagues and medical students and other things. I do some seminars in immunology sometimes. Other than that, not much.
Okay. Now you teach virology? Yeah. An entire course or part of another?
Well, it's part of a course in microbiology, and it's also integrated with pharmacology, which is it's almost like two courses. And So I primarily teach the medical students, and the bulk of it is in their 4th semester. And in the first half of the semester, they have microbiology, and I teach all of the virology, or almost all. Matti, who you met in the morning, he's the only one. He comes in and gives 2 lectures. I give 20. And I also, I'm responsible for their seminars, where Anna and others help out with teaching. And then I also teach later in the 4th semester when they do clinical microbiology. So that's more sort of what samples are needed, what tests are run, and how should you think when you have a patient with these symptoms when it comes to microbiology testing. And it's great fun. I like teaching. It's kind of challenging because the classes here are very big. It's 160 to 180 students per semester, and up until the 4th semester, they are all together. So most of the teaching is to this really big group, which is a challenge. The seminars are of course in smaller groups, and then the students split up at different hospitals in Stockholm.
So how do you make virology interesting for a medical student?
Well, I try to remember that they are not primarily going to be virology or microbiology researchers, but they're going to be some type of doctor. So I think the fact that I'm a medical doctor and I've practiced medicine. I try to remember that and always connect to sort of some type of reality and not go too much into details, which may be super interesting for me, but maybe not so much for the students. And give sort of examples of recent things, things from the newspapers, things like that. Try to make them feel that microbiology is super fun and interesting, and even if they won't practice it, they should remember that it's fun and important.
And so they like it in general, right?
I think the fact that it's slightly different. The approach that I've taken, that I give all of the lectures myself, usually gets good reviews, and I think the main reason is that I know what I've said in the previous lecture, and I know what I'm going to say in the next lecture the next day, so I can connect and I can keep like some type of continuity, and I have a plan for the entire thing.
Yeah. So students in general don't like team-taught courses where you have a lot of people teaching it. Yeah. They like have one person. Now, if that one person is not good, then
that's sort of the challenge.
Problem.
But these plenary lectures are not compulsory. So the ones that don't like me, they don't come.
Well, if someone is not a good teacher, they're not going to do 20 lectures. At some point, they're going to stop teaching. I teach undergraduates. I teach 25 lectures myself for the same reason, because I know how I want to do it, and there I don't have to worry too much. Even though half of my class is pre-med, I don't have to make it medically relevant. I teach them basic science. I say, you all have to know this, and as long as you're passionate and excited, I find it goes a long way. Right.
Yeah, I think so too. I try to convey that I think that this is really fun.
Also, if you can bring in contemporary things, and you can say, "Oh, look what we found today in the lab." Some stuff like that is really good, I think. Right. All right. I have one more question, Anna. If you had not been a scientist, what would you have done in your career?
I think I would have become a musician, most likely. I also did a parallel sort of music training and was,
What instrument?
Well, I played the recorder, so I played Baroque music primarily since I was 5.
And you still do?
Oh, less so now, I would say, but I did intensely for many years.
What about you, Petter? If you hadn't been a physician scientist,
I don't know. Physician only, or
that would be okay.
maybe a professional hockey player. I don't know.
Were you good at hockey?
I was pretty decent, yeah. But even though I'm pretty small, but I was pretty fast.
So I was talking to Erling Norby last night, and he said one of the cool things about science is you can go to different places. And he said he once went to a hockey game in Edmonton and saw Wayne Gretzky. He thought that was very exciting.
Well, yeah, who wouldn't? No, I don't know. Yeah, maybe.
But you wouldn't have stayed in the military, right?
No. That was we had a military service that it wasn't really compulsory, but almost, I guess, when I was 19, 20. So everyone did something. It wasn't really a choice, yeah.
And what about you? If you hadn't been a physician scientist, what would you have done?
I would probably have been in something related. I guess I was very much interested in school, in math, chemistry, physics, things like that. So that's the most likely. I like music, but I wouldn't have become a musician. And I played hockey, but I wasn't good.
Maybe you would have taught.
Maybe, but it's not something that I really aspired to be. It was more or less forced on me. But then when I started doing it, I liked it, and I wanted to do something good of it.
Yeah.
So it was this sort of relatively large responsibility for teaching is not something that I've had for years and years and years. But still some years, because I'm sort of senior. So now probably not teaching.
Okay, I got it. Anna, RNA or DNA?
RNA.
Patrick?
RNA, definitely.
Wow, I never had all RNA. I always have a mix. That's great. All right, that's TWiV, a special TWiV here in Stockholm, number 2 of 3. I'm doing 3 TWiVs in this country. I think that's the most I've done anywhere except for the US, so that's fun. You can find it on any podcast player, on your phone or tablet. If you listen, please subscribe so we know how many people are listening. And if you really like what we do, consider supporting us financially. You can go to microbe.tv/contribute. And I have to say, the whole idea of getting listeners to contribute was really Kim Vesto's idea, who is one of my 2 hosts here in Stockholm. He arranged this particular TWiV. He's been a listener for many years. And a couple of years ago, he said, you should get listeners to contribute. So I said, oh, yeah, you're right. And so I did. And so now please contribute because of Kim. Microbe.tv/contribute. You could give $1 a month, for example, and that would help us with our expenses. My guests today, all here at the Karolinska Institute. I don't know why I can't pronounce that properly. Jan Albert, thank you so much.
Thank you
for joining me. Petter Brodin, thank you.
Thank you very much.
And Anna Smed-Sörensen, thank you.
Thank you
so much for joining me. I'm Vincent Rackaniello. You can find me at virology.ws. I want to thank Kim Vesto for organizing the TWiV and showing us around this campus, the cool Nobel-related places. I want to thank ASV and ASM for their support. Ronald Jenkins for his music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.